another story, something to get off my chest.
Hello and welcome to another Piper Pod. This pod will finish the Anatomy Physiology series before we move on to something else. So today we're going to talk about protein synthesis. So we're going to go through the importance of protein in the body, how it's formed, how the different structures of proteins are actually made, and how it's transported around the cells and throughout the body. So first of all, let's look at all the different things proteins are used for. Now when we look at a protein chain or a polypeptide, it often follows the same pathway, or the same travelling pathway at least, as your lipoproteins as well. So when you say proteins, we can have lipid or steroid-based substances that are mixed in within it in order to make a functional unit. So all of your hardened areas around your body, the keratin of your hands, your hands, your nails and your hair are made by protein structures. Hemoglobin is one of the classic ones where protein synthesis happens en masse within a stem cell. The hemocytoblast will actually go through the production of hemoglobin tenfold before it dumps everything else except its hemoglobin chains in order to become red blood cells. Any ion channels that we speak about when we deal with nerves, brain activities, sarcoplasms, every single one of those channels is made up of proteins themselves. And then you also have a series of messengers. So your enzymes, your hormones, your receptor mediators, your coenzymes, your cytokines, every one of those is made up of these protein chains. As well as that, obviously your muscle structures are constantly repaired by protein chains. Okay, the very small actin and myosin are themselves just specialized protein chains. And your cellular immunity, or humoral immunity as well, is all made up of your protein chains. So as you can see, it's a very useful substance to have, and what you'll find is every cell with a nucleus is responsible for the production of some sort of protein for the body. So a protein chain, or proteins themselves, are made up of small building blocks called amino acids. Now there's 20 different amino acids that the body utilizes, and each cell needs a combination of each one of those 20 amino acids. So remember, when we talk about metabolism of protein, why the body doesn't want to use it and only will when there's abundance or starvation, it's because every cell has to have a mixture of these amino acids in order to produce the different protein molecules. So these amino acids come from the food we eat, and it's also some of them can be synthesized from the body, from breaking down other substances that we eat, but essentially it comes from our diet and it's transferred around the body in order to be stored in every cell to be utilized. So when we say a protein molecule or a polypeptide, just think that it individually is made up of these amino acids. Okay, getting right into it now. Now that we know the background of what protein's used and how it's made up, let's talk about how the recipe or how the construction of these proteins actually goes. So a lot of people like to talk about the cell and like to refer to them as different aspects of a city, if you will. So when everyone talks about a mitochondria, the main thing people remember from primary school, whenever it was, is that was the powerhouse. Okay, so during this protein synthesis, I'll try to draw connections using similar analogies and attempt to make it a little bit more obvious and easy to remember. So right at the start, within the nucleus, which can best be described as company headquarters, where the orders are coming from for blueprints in which factories can make. So obviously the nucleus is where the DNA is housed. Now last lesson during mitosis we talked about DNA synthesis. This time we're going to talk about the production of messenger RNA and how that slightly differs. So within a cell, as you know, each cell has its own specialized structure to produce different sorts of proteins, and that is the part of the DNA that is going to be open and active, so not wrapped around those helicase that we spoke about. So in the event that we need a particular protein made, helicase will come along and will unzip that component of the DNA. From there, we'll get our based nucleic pairs to bind against it, but this time, instead of adenine binding with thymine, adenine will bind with uracil. And that's what makes the difference between DNA and this complementary messenger RNA. So the other nucleotides will line up together, guanine with cytosine, adenine uracil, thymine with adenine, and cytosine with guanine. Each of those will base together and it will create its own messenger RNA chain that can move out of the nucleus itself. When the messenger RNA transfers out of the nucleus, it is the company headquarters sending orders out to the factory. Now, when it leaves the nucleus, it enters an aspect of the cell called the endoplasmic reticulum. Now, the endoplasmic reticulum is the highway of the city. It is the internal highway in which the message can be transferred around to the different factories in order to produce polypeptides. Now, the factories in this case are called ribosomes. Now, ribosomes sit all the way along the rough endoplasmic reticulum. And when the messenger RNA gets to the ribosome, it goes through a process of decoding. Decoding is a way of saying that it's reading the blueprint and then connecting the complementary nucleotides to the actual blueprint. Now, the way this helps with protein synthesis is each base nucleotide is read in a series of 3. So 3 nucleotides have a complementary codon. Now, a codon is transferred towards the ribosome to read the messenger RNA by a specialized transfer RNA. So each transfer RNA has a complementary codon to read the messenger RNA. So as you can see, the triple uracil transfer RNA coming in is going to bind to the triple adenosine messenger RNA. Now, on top of the transfer RNA is a complementary amino acid. And it's these amino acids that will eventually form the protein chain. Now, each of these 3 nucleotides attached to the codon is specifically designed to carry a certain amino acid. Now, this picture here looks a little bit confusing at the start, but if we just break down the actual trinucleotide base pairs, then you'll see that they link nicely with a particular amino acid. So looking at the code above, you've got your adenosine, uracil, and guanine. You go down to the map, you start at the bottom left corner with the adenosine. Moving along to the uracil, and then into the guanine, you obviously have your methylene right there. And that continues along, so each of the trinucleic group, or the codon, will bind to the transfer RNA, which is read onto the messenger RNA that brings along a specific amino acid. Now, as you can see at the end, uracil, adenosine, that goes with a stop code. Now, the advantage of having a stop code is you can actually have one messenger RNA that can do a series of different polypeptides. So instead of having a recipe for one, you can just put a stop in the middle, and that breaks the chain, so you have one peptide before it starts on the next side of the house. So once the polypeptides start lining up and are being transferred by the transfer RNA, Then the polypeptides break their bond with the transfer RNA and then actually start to bind together. The polypeptide will actually have a bond together and break off the transfer RNA in order to create a chain. Now the transfer RNA will simply move off the ribosome and travel back towards the nucleus in order to bind with another amino acid for the next production. By the end of it, what we have is a long series of amino acids called polypeptides. Now polypeptides are the starting point of what we commonly call a protein. Now remember, all this is done on the ribosome, which is within the rough endoplasmic reticulum. Now from there, all we have is a long protein chain, a really long series of amino acids that are just in a long chain, which is not what a protein looks like. Protein looks like the pictures in front of you here. Now the way in which a protein molecule is formed from a polypeptide is you need to imagine that within that polypeptide we have a series of different amino acid groups all along the chain in specific orders. Now what happens in order for a protein to form its shape and twist a certain way in order to have different receptor sites or binding actions within it is the amino acids, or the linked amino acids, meaning methylloin and methylloin, will twist the actual polypeptide chain. So methylloin and methylloin are bound together, lysine and lysine bound together, and all the other amino acids are twisted so they are sitting next to their complementary amino acid. Now by doing that, you get a long chain that is twisted and manipulated to create a different substance or a different structure. So as you can see, when we look at the classic hemoglobin, we have a cluster of different colours all together. Now those colours represent the same amino acid within that particular hormone, and they've twisted around themselves in order to be close together. And that's what gives it its shape, and that's what differentiates the different protein structures and allows them to have different functions, because all they are is a series of different amino acids all stuck together. So in that state, they're completely useless. It's only when they're twisted and manipulated and given form that they can actually function. Now, on a clinical side note, this is the reason why when we have states of acidosis, hypo or hyperthermia, their protein molecule substances like hormones, enzymes cease to function. So enzyme activities can't occur in acidosis because this tightly bound and twisted perfect shape designed for function starts to degrade and move apart. Therefore, the binding or active sites within the enzyme are no longer viable. The heme group within the hemoglobin is no longer able to bind to oxygen because it's been altered by the acidosis or by the heat or the cold of the body. So that's how protein masses or protein molecules are formed from those amino acids. From there we have a protein within a cell. Now obviously there's a lot of proteins out there that are pretty useless within the cell, so they have to try to get out of the cell. So the picture in front of us is a very zoomed in version of the cytoplasm against the cell membrane. So the large structure at the bottom represents the endoplasmic reticulum, and the little dots on it is the ribosomes. So once that protein molecule is created, and it's looking pretty good, it's starting of a polypeptide, it starts to form into a protein molecule, it is transported from the endoplasmic reticulum to the Golgi apparatus. Now the Golgi apparatus can best be described as the cell's harbour. It's responsible for packaging and receiving different packages for transport in order to bring into the cell or export out of the cell. So that's what the Golgi apparatus does. So this long polypeptide chain will enter the Golgi apparatus where it is fine-tuned, where it goes through its manipulation of twisting and bending and being formed into a proper protein molecule. From there, it is wrapped in what's known as a vesicle, and it is transported towards the cell membrane itself. Now, the process of pushing a vesicle with a protein molecule within it towards the cell membrane to be put out into the body is called exocytosis. Now, the way that works is the vesicle will fuse with the cell membrane, open it up, create a hole, and then essentially push out the actual substance within it, so in this case the protein molecules, and then close back over the actual cell wall itself. In reverse endocytosis that we've talked about, this is where we bring molecules in or substances into the cell. Now that is done by gathering it all into the cell membrane, and then the cell membrane itself will close over this group, creating endocytic vesicle. Now that vesicle itself can move down, it can go towards the Golgi apparatus in order to be processed into the cell before it is transferred back to the endoplasmic reticulum to get to the area of the cell that it needs to be in order to function. A different thing that it can actually do is move into an area creating lysomes for destruction. Now this is sort of what happens when we're dealing with phagocytosis, which we talked about before. It brings in the substances and then it injects the lysomes in order to destroy it before it goes through exocytosis out the other side to get rid of. Thus completing the entire process of protein synthesis, all the way from blueprint recipe from headquarters into manufacturing at the ribosomes and exporting within the Golgi apparatus. This process happens, as I said, in every cell that contains a nucleus, and as a bit of a sidebar, this is the exact process that viruses exploit within our cells in order to reproduce itself. A virus will actually inject its own special DNA into the nucleus in order to be copied as messenger RNA that will go through the exact process I just talked about in order to generate itself on the inside of the cell until it has large enough in numbers to cause the cell to erupt, to transfer to other cells in order to go through the same process. Okay, so nice short one, finishing off anatomy physiology. I'm not sure what's coming next after these, but until next time, take care.